to talk about interesting topics. Today, we have Dr. Chidozie Ojibor. Am I saying that right?
Yeah.
Close enough, maybe, who's going to talk to us about the microbiome. And I think what we can all relate to is how important the microbiome is. I think we all intuitively know that for health, and yet it can feel difficult to wrap our heads around how best to apply it for patients. Perhaps some of us have experience ordering tests like the GI map and others, and I think we can all relate to getting these test results back and sometimes feeling like I'm really excited to have Dr. Ojibor share with us how he thinks about the microbiome, and I think this is going to be a very useful perspective. Finally, I would say if any of you guys are interested who are not already part of the program, interested in joining, please send me an email and I'd be happy to talk through that with you. All right, with all that being said, floor is yours.
Thank you very much, Anant. I know you said that there's different approaches you take to this, whether it's conversational or people just wanting to ask questions. I'm happy if you just want to engage me. I'm happy if you just bombard me with questions. That's
Yeah,
fine.
so what we can do is I can start by asking you a few questions, and then from there, if we have time, I'd love to open it up to the group. So
Okay.
the title of this webinar was a first principles approach to the microbiome, which I think is important to take to really any topic. Like I'm a big advocate of a first principles based thinking where we think about things through the lens of the fundamental principles that inform how something works and then use that to inform how we apply it. So I guess when it comes to the microbiome, what are some of the key frameworks that you use to understand how the microbiome is relevant to clinical application?
Absolutely. Before I get into that, I'll just quickly introduce myself. Your community can at least know who I am. I actually started my research in core microbiology, where I got really fascinated about viruses that cause gastroenteritis in children. And so I started with really looking into rotaviruses. That was the first time I started collecting stool samples. I think I collected about 180 to 200 stool samples over 2 years. And we're just looking out for the specific genotypes and electropherotypes of those rotaviruses that cause gastroenteritis. That was a fascinating project. But I think where I started getting even more interested in the gut microbiome was during my doctoral studies at the University of Toronto. There, I was developing novel therapeutic tools against the drug-resistant bacteria. They're called bacteriocins. So when you look at these bacteriocins, they're macromolecules that look like bacteriophages, which are viruses that infect bacteria. But the really interesting thing about them is they have a very narrow host range specificity. These are macromolecules produced by some strains of bacteria to target other strains living within that same ecological niche. They just have strain at most species specificity. And I found them really interesting. When you talk about antibiotics, one of the hugest impacts on people's gut microbiomes today is the use of antibiotics because they have off-target effects. But with bacteriocids, because of that narrow specificity, I started to think about them as biological tools to manipulate the gut microbiome in the sense that if I'm interested in just this one organism and I want to target that organism, what biological tool can I throw into such a complex biological ecosystem and ensure that I'm targeting the organism that I'm interested in and not having off-target effect? So to your question, when I think about frameworks guiding microbiome, especially in relation to clinical application, I start with trying to address one of the most fundamental recurring lines I've heard, whether it's me talking to scientists or talking to average people who are interested in the microbiome or even clinicians. It is that the microbiome, we do not know what is composed in a healthy microbiome. So how can we look at such a complex thing and then think about how we can apply them to clinical practice? And I think it's a really fundamental and valid argument because two healthy individuals can actually have different taxonomic profiles. That is to say that Anant and I might be healthy, but when we do a microbiome test, we're definitely not going to have the same strains. At best, we'll probably share 30 to 40% of the same strains. So how do you make a clinical call from something that is that diverse? I think I would like to start from there because I do think that the framework guiding microbiome understanding in relation to clinical application stems from here. So I call that microbiome bioindividuality, a situation whereby two individuals might have different gut microbiome profiles, yet are both healthy. Now, that's because we do know that although these two individuals might have different taxonomic profiles, they look alike more when you start looking at them at the functional level. An ant might have a dominance of organism A, B, and C, and I might have the dominance of organism D, E, and F. But those combinations of organisms are all producing beneficial metabolites like B vitamins, or they're producing neurotransmitters. So at the functional level, an antibody might look similar, but at taxonomic level, we might not look so similar. Which is why when you're looking at the microbiome, what we start to look out for now, the conversation has gone beyond what are the organisms that make up a healthy microbiome to what are the signatures and the fingerprints that actually underlie health and disease. And so that's where my framework would start. Now, and because I've looked at thousands of microbiome data now with corresponding clinical metadata, it allows me that opportunity to actually put this in context because now I'm not just looking at them in the context of people's clinical data and microbiome profiles, but a lot of this data have also been
Well, thanks so
constituted.
much.
So the first thing I will look out for is one of the things that underlies health is we have seen what we call gut diversity to be associated with health. A lot of us here have heard, oh, eat more rainbow diet just to increase the number of species in your gut ecosystem. More often than not, the more species you have in your gut, the more likely you are to achieve health. The question is why? Because I think when we talk about first principle approach, you must always ask the question why. Like, why is it that more number of species has been linked in more studies than not to health? It is because of what I call the functional redundancy of the microbiome. So if Anant and I took a gut microbiome test today, Anant has 500 species and I have 200 species. If we do this across different populations with disease conditions and a well-characterized healthy control, we would see that the more species you have, the more likely you are to achieve health. But why? The reason is we always gravitate towards having more species to increase the tendency of more than one organism producing the same metabolite. That's functional redundancy. So if I have 100 species and only one or two of those organisms are able to produce butyrate, what would happen to me if I start taking antibiotics or if I have my IBD flare up? There is a tendency that one or two organisms that actually produce that beneficial metabolite is wiped out. And that then decreases or impacts negatively my microbiome health. But if an ant has 500 species and 6 or 7 of these organisms have the biosynthetic pathways or encode genes that can produce this same metabolite, then in the case where one or two of those organisms are knocked down, the other functional lookalikes can come in and set the stage for improving your microbiome health. So that's where I usually would start from. One is improved gut diversity associated with health. The other thing that we have seen that is quite common in people that are healthy, unfortunately, it's very difficult to define what healthy is, especially in North America, where almost everybody has some sort of low-grade inflammation. Let's talk about healthy in the context of not presenting anything actively, you don't have any diagnosis and things like that. I know a lot of you here are clinicians, so you probably would better define health here. But the second thing that we tend to see in this group of people that are healthy is we tend to see a dominance of what we call the beneficial probiotic strains. Now, these organisms do three key things. One, they help to improve the gut barrier integrity. And they do this by helping to, whether they help to increase the thickness of the mucus layer. We see this with organisms like Akkermansia and Muciniphila. I mean, from the name, Muciniphila, it's mucin loving. So these are usually mucosal adherent organisms. They feed on the mucus lining, on that mucus. But on the by feeding on that mucus, they produce acetate. Now, acetates then incite your goblet cells to produce more mucin. So there is that degradation of mucin that also incites the synthesis of more mucin. And you want to see a homeostatic balance there. You do not want to see excessive degradation of the mucin layer, as we tend to see in some individuals actually. Unfortunately, Akkermansia is sold by a lot of commercial probiotic companies that promoted. We have seen people that have excessive Akkermansia and are dealing with a lot of inflammation. And that's not unsurprising because we're just having far too much degradation versus synthesis. So that's what the first thing these probiotic strains do. The second thing they do is they produce beneficial metabolites, such as the short-chain fatty acids. A very key example here is butyrate. Butyrate is powerful because most of these organisms can produce butyrate from the fermentation of dietary fiber. We tend to see these organisms cross-feed on other short-chain fatty acids like acetate to produce butyrate. A key example here is Faecalibacterium proteinaceae. Most organisms that produce butyrate and other short-chain fatty acids typically are obligate anaerobes. That is to say that they will rather exist in the absence of oxygen. All right. And what we tend to see here is that even when you introduce a very low amount of concentration of oxygen at microaerophilic levels, we tend to see a reduction in some of these beneficial organisms. Now, butyrate, we know, is the primary source of energy for your colonocytes. 70 to 80% of the energy that your colonocyte uses come from butyrate. So rather than take the band-aid approach to maybe supplementing with just sodium butyrate or hydroxybutyrate, we have to start thinking about ways we can actually increase the number of strains in the gut microenvironment that can produce this butyrate. And there's a lot of them. There's the Faecalibacterium, there's the Rosaburias, there's some strains of Bifidobacterium, there's Anaerobutyricum heli. And I think that where the conversation would go is, how do we think of modifiers, which are the foods, the supplements, the prebiotics that can actually help to increase these strains? So that's the second thing from that first approach perspective that I think of. But then the third thing here is, I think probiotics strains are organisms that have been shown, and this has been recapitulated across different studies, to actually help create an anti-inflammatory state in the gut. Well, they do this by maybe dampening the Th1, Th17 bias, and they do this by helping to prevent that translocation of lipopolysaccharides from the gut lumen into circulation, things we know that can actually drive a very complex immunological state. That's the second thing. Like I said, I know there are 3 or 4 frameworks. Finally, we tend to see very low levels. These pathobionts, although we see them in healthy individuals, we do know that they exist in very low amounts. Now, when there is some sort of dysbiosis or imbalance, maybe you just got out of a round of taking a lot of antibiotics, what we tend to see is that it leaves room for some of these pathobionts to actually bloom. A key example of these pathobionts are proteobacteria. It's a phylum of your bugs called the proteobacteria. This comprises about over 140 species. I worked with some proteobacteria during my PhD. What I can tell you here is one of the most fascinating features of this proteobacteria is on the outer membrane of these organisms, they have lipopolysaccharides, which is a lipid that is bound by repeating units of sugars. Now, these lipopolysaccharides are highly pro-inflammatory. We were doing an internal analysis recently where we were looking at the gut microbiome profiles of people that had insulin resistance and type 2 diabetes. Comparing them to a well-characterized healthy control, one of the key things we could see was a significant difference in the levels of proteobacteria. So we're not even looking at the microbiome at the functional level, which is the level that gives us more insight. We're talking about the taxonomic profiling level. And in healthy individuals, we see proteobacteria to be around less than 5%, max between 5 and 10%, max. But we tend to see in these people that have reported insulin resistance, T2D, even as high as 30%. Now, what does that tell us? That gives us an insight into what the nature of the microenvironment is. Proteobacteria are organisms that can live in the presence of oxygen. So if the luminal microenvironment of your gut is not hypoxic, you cannot enhance the growth of beneficial species like Faecalibacterium, but you're going to enhance the growth of Enterobacteriaceae and Proteobacteria. So that's one, which is why I think we should start redefining what dysbiosis is beyond imbalance in the bugs themselves, but we have to also think about what the microenvironment is. Secondly, what these proteobacteria we think they're doing from a hypothesis point of view is we think that when you have leaky gut and some of these lipopolysaccharides that are born on the outer membrane of this proteobacteria escape out of the gut lumen into circulation, we think that they have the potential to impair glucose metabolism. We've seen this actually happen in some studies, for example, in the liver, which contributes to why people with type 2 diabetes would most likely have elevated levels of proteobacteria. This is how I think and about that first approach and principle of microbiome analysis.
Got it. That was tremendously insightful and revealing. And I hope everyone listening appreciate just how important it is to understand things from a first principles perspective. What you pointed out goes against how this information is presented by some of the testing companies, where there's just like these red and green bars and you want everything green. It doesn't really tell you what actually is going on. And I think there's a couple things I want to highlight for the group that you mentioned, right? Number one is the importance of the functional products of the microbiome. I think there's maybe too much emphasis on good and bad bacteria. As he pointed out, that type of framework is not correct. It's not consistent with how things actually work. We've all heard Akkermansia is a great example, where like we're bombarded by vendors who are pushing Akkermansia and being like, oh, take more Akkermansia, it's good for you. And it certainly can be, but I think we have to understand, what is it doing for us? What is the functional end product of Akkermansia? How does that functional end product spectrum look for a given patient? And so I think that's one very important thing to understand, is like the importance of the functional end products. The other thing that was new to me was this idea of why diversity matters, right? We're told, oh, you got to have a diverse microbiome, eat the rainbow, have all these things. But why does that matter? What you pointed out was it's because you need backups for producing these important metabolites. And I think that's why diversity matters. We should be moving to a more functional model of the microbiome versus this simplistic taxonomy. Here's a species, here's the good ones, here's the bad ones, and you want more of the good ones and none of the bad ones. I feel like that's maybe not a useful frame. I guess just to ask you about one concept that at least I was taught about was this ratio, the Firmicutes to Bacteroides ratio. What do you think about ratios like that? Are those based on the same kind of flawed thinking?
Yeah, I think a lot of those ratios are now outdated. When we started learning more about the microbiome, we started to see some interesting signatures that were correlating to ratios. And remember, the primary technology used to probe the gut microbiome was the 16S ribosomal RNA sequencing, where you can infer what an organism is based on one gene. In fact, not the whole gene, just a very hypervariable region of that gene. It's like you're looking at a Ford car logo, and you can tell that the car is Ford, but you can't tell if it's a Ford Escape or a Ford Fusion or Ford Focus. So you don't have that level of resolution. When we started to understand the microbiome from that angle, there was a lot of really interesting things, and one of them was the bacteroides ratio. In fact, one condition that ratio was associated with was obesity. And every single day, as papers are being published, we're seeing that model doesn't stand. Because we're looking at two different phyla here, make up in healthy gut ecosystems around 95%. But that ratio could go from 10% to 90%. We've seen people have just 20% bacteroides, 70% firmicutes, and the other way around. But collectively, they make up a huge part of your gut microbiome. We've also seen healthy groups, regardless of whether you have 10% or 60%. In fact, for obesity, it was interesting because at the time when that ratio was a thing, studies were almost completely defining for obesity. For example, you look at obese individuals and take the stool samples and you transplant that into mice, and that mice became obese.
Hmm.
And those are some of the studies that actually, it means that we could actually have an obesogenic microbiome. But as we started learning about the microbiome from different groups and populations, we saw that we could not recapitulate or reproduce that data. But in terms of ratio, there's some ratios now that seem to give us more insight. An example would be the fusobacterium to the fecalibacterium ratio. This is why I believe that one of the key spaces that would see very rapid application of the microbiome might be in oncology, because now we are beginning to see how we can use specific microbiome markers that can give us an insight into conditions like colorectal cancer. Bacterium nucleatum is one of those organisms. You can increase the sensitivity of diagnosing colorectal cancer with FIT. You can increase the sensitivity by 20% if you paired FIT with a microbiome marker such as fusobacterium. With fusobacterium alone, sensitivity is about 80%. So 8 out of 10 times you'll catch a true positive, especially when you're comparing against a well-characterized control. Now, there are studies that have come out of China, very more than, I'm talking about 5 to 10 studies now, been done by different groups, where you can use the ratio of fusobacterium, a very nasty organism that is highly pro-inflammatory. We think about them as either drivers of the inflammation or passengers of the inflammation. If the inflammation is already there, they're going to further drive it. We've seen them to be super enriched in people that have advanced adenomas and colorectal cancer. And we have seen a concomitant reduction in the levels of Faecalibacterium proteinaceae, it's obligate anaerobe, that's the largest producer in the gut. So that ratio seems to be a really interesting ratio in the oncology space right now. And I'm really looking forward to how that ratio can be recapitulated by more studies, especially when we look at the Western population. I think those are some of the interesting ratios that I'll be looking at now because they are also more specific.
Got it. Thank you for that. And I guess just to make this super concrete for the people listening, we've talked about some of the first principles based approaches to thinking about the microbiome. We've talked about some of the issues with how it's often presented to us. What are some practical ways to actually engage with the microbiome clinically? You can feel free to talk about your test that you work on.
Yeah, absolutely. There are different ways to get insights into microbiome. And I'm happy to talk through most of these techniques. First, we started with culturing, just trying to understand what's happening in the gut by plating those organisms, taking your stool sample and culturing them on the plate, growing them in a 37-degree incubator, and seeing what grows. I did that 15, 20 years ago in classic microbiology. Some of you here who are clinicians probably also did that. But now we know that what can grow in the presence of oxygen is about 15% in your gut. So if you stick to culturing, you're going to be losing out information on about 85 to 90% of the bugs that are growing in your gut ecosystem. So that's one. Two, one of the cons of using culturing as well is that there are organisms, because all you have done is you are isolating an organism and growing them on a petri dish that has a media likely to grow one organism more than the other. So what you're going to be seeing on the plate would not be what's actually happening in a complex biological
And
ecosystem.
just to be clear, do most of the tests that physicians order today use culture? Is that accurate?
Some of them still do. Most of the tests are next generation genomic based testing, like PCR and 16S and metagenomics. But we still see some legacy tests still have culturing as one panel there where you see 4 plus, 2 plus, 3 plus, and then they do some sensitivity tests on them. It's not very informative in the context of the gut microbiome.
Got it.
The other technique, we went from culturing to 16S, which is something I described just shortly, where you can identify an organism just by looking at one gene. Everyone in the microbiome space got super excited about 16S because even for the American Gut Project, the F Microbiome Project, all of these big projects, 16S was actually used to probe our gut ecosystem. It allows us to have a more expansive view of what's happening in the gut, but the resolution is low in the sense that we can only identify these organisms down to genus level. That is to say that there are other levels that we can identify these organisms at that 16S does not have the bandwidth to do. For example, the species level and the strain level. And we know, of course, that even at that species level, there's a lot of genetic diversity. So 16S is another way to actually probe the microbiome. My company, which I started 3 years ago, launched our first product about 1.5 years ago. Our first product uses RNA sequencing to probe the gut microbiome. Now, the other technique to use is metagenomics, which a lot of us call shotgun sequencing. With shotgun sequencing, you identify an organism to strain level because the approach we're taking is whole genome sequencing. We get your stool sample, extract the DNA, split the whole DNA, and assemble them. That way, we are able to identify an organism down to strain level. Why is this important? We know that at strain level, there's a lot of diversity. Take, for example, E. coli. We have some E. coli strains that have 4 million base pairs. Their genome is made up of 4 million base pairs. And we have other E. coli strains whose genomes are made up of 6 million base pairs. And that is enough to actually make one super pathogenic and one a commensal or even a probiotic. And that's why there's some probiotics that are E. coli strains, but of course we have enteropathogenic E. coli as well. So with metagenomics, we can drill down to that specificity. Another company, I know they use metatranscriptomics, which is where they give you an insight into the gene profile, that is the RNA, they measure the RNA levels of all the communities in your gut ecosystem. And that's a great technology. It's an emerging technology, but it's absolutely not a good technology to use for routine microbiome test. And this is why. There's a couple of reasons. One is RNAs, as some of you here know, are very unstable, right? Because of how unstable and how transient RNAs are, you need extra care in the lab to ensure that you're preserving the RNA. But I think that's even the least of the problems. The other problem is we have also seen that because what we're collecting in that moment is the real-time expression, it does not necessarily give you a sense of what is actually happening in that gut ecosystem. It just tells you what's happening now. And that's why in several studies, we have seen a very big mismatch between RNA profile and the eventual protein profile, because eventually these RNAs have to be translated into proteins. And there are several studies that have shown that when you look at the RNA profile from the same sample and you look at the protein profiles, not all of those RNAs eventually make enzymes. Not all of those RNAs are transcribed to proteins. These are the four fundamental tests that have been used to try to really understand the microbiome. An emerging technology now is a lot of these metabolomic approaches, and now we're using them in microbiome work where you're doing things like gas chromatography. Here we're measuring the non-protein metabolites themselves. So instead of just measuring the species that produce the butyrate, can we actually measure the butyrate in the gut ecosystem? The sweet spot is where we can combine metagenomics that gives us a true sense of the genes that are encoded down to strain level, metabolomics, where we can actually measure the non-protein metabolite. I think these two technologies paired together is going to be the future of the microbiome, because now we can truly understand things beyond taxonomic level, beyond functional potential level, to actually having an insight into true footprint of that community in the gut ecosystem.
Got it. So what's the best way for us to do that now? And how, if we were to order a test, what are some markers that we should look for? Like, how would you guide us to interpret any microbiome tests that we get?
Absolutely. So I'll use our test as an example. What we have done is we have what we call a deep gut test, which is a deep shotgun sequencing. So it's a metagenomics test that helps to identify things down to strain level. Here we have the first section where we give you an insight into diversity. That is the total number of species and how evenly spread out they are. We've talked about why we need high diversity. So that's the first thing. We give you a clear picture there. And we compare you to a cohort of 1,071 healthy individuals. All right. So that's the first thing. The second thing we show you in the report is a sense of the organisms that have been researched on and reproduced by different groups to impact health and disease. It's not just about this organism is bad and this organism is good. We put that in context for you. What are the organisms associated with mucosal health? What are the organisms that are associated with short-chain fatty acid production? What are the organisms associated? What are the keystone species? Now, we're also able to identify what we call pathobionts, like these organisms that are producing a lot of hydrogen gas or hydrogen sulfide or producing a lot of methane. So we stratify those organisms based off on their functional potential. The third thing that we show you is we show you the functional ability of your gut microbiome based off on your composition. Do you have enough strains that are producing short-chain fatty acids? We know that your gut microbiome is one of the largest sources of your B vitamins. Are you producing sufficient B vitamins? And if you're producing them, what type of B vitamins are you producing? B9, for example, about 50 to 60% of your gut microbes actually have genes encoded that can enable them to produce these B9 vitamins. B12, about 30%. So we give you that insight. There are some longevity markers we are also able to show, things like myrosinase, which can only be produced by your gut. They help to break down specific substrates into compounds like sulforaphane that have antioxidant properties and anti-cancer properties and things like that. So we give you that functional insight. But the last thing, which I believe every microbiome company should really strive towards, and this is one of the places that we've spent a lot of time innovating in, is the actionability of the test. At the end of the day, the question people ask you after doing a microbiome test is, what should I do? Regardless of this is high, this is low, what should I do? So the way we thought about what to do is we stratified what you can do into 5 different categories: foods, prebiotics, probiotics, supplements, and lifestyle changes. It's a very rigorous task to actually recommend a single, whether it's a probiotic or food for the microbiome, because again, it's a complex system, because there are different groups that have done different things. Some of those studies actually conflict. So we have set some rules to identify the modifiers, the foods that are more likely to shift your gut microbiome. First, we prioritize studies that have been done in humans. We prioritize studies that have been done in different populations. We prioritize interventional studies, all right? Versus we don't give the same weights to an interventional study versus a cross-sectional observation study. So these are the ways to actually look out for specific things in a gut microbiome test. And lastly, as a clinician, if you order a test, what should you be looking out for? First, you should be looking out for gut diversity. How diverse is this person's gut? What are the total number of species that are there? How evenly spread out are they? The second thing you should be looking out for is, am I seeing sufficient species that can do 3 key things? Sufficient species that can help to repair the gut lining. That is, are they enhancing the production of mucin? That could be by production of more GLP-1, which we know is associated with also that repair of the gut lining, or more acetic. Secondly is, do we have enough species that are producing enough short-chain fatty acids like your butyrate, your acetate, your propionate? And thirdly is, do we have sufficient organisms that can help to keep an anti-inflammatory state? That's the other thing you want to look out for. And then you also want to look out for organisms that can potentially be pathogenic. They are opportunistic pathogens. I must mention here, most of these organisms, we see them in healthy people. So the fact that you saw maybe a biolophila does not mean that the person has a problem. But we try to show you the ratio of what we typically see in healthy groups. These are the hydrogen sulfide producers. These are the methane producers. We have a lot of people who are dealing with IBS and IBDs. One of the things that we've done is we have integrated into clinics directly versus like sell direct to consumer. By integrating into clinics, we can collect a lot of metadata that allows us to have more insight. We have seen, for example, people who are dealing with diarrhea predominant IBS. From our internal analysis, we have seen that there are specific species that tend to be enriched that you can use to distinguish diarrhea predominant IBS versus constipation predominant IBS. So we find them, we report them, and we show you in ways that are easy to understand and that are very actionable.
Thank you for that. I now want to turn to some questions that have come through in the chat. One thing that is implicit in both of the questions that I'll ask explicitly is, how would you compare the different microbiome tests that are out there? And I think just to name a few, there's the GI map, the GI zoomer, the gut zoomer from Vibrant. There's your test. There's Jonah. There's Biome. How would you characterize these? How would you put them into buckets? What are the strengths and weaknesses of each?
Yeah. Okay. So I'll separate those companies into 3 main groups: GI map, Godzoomer and Vitract, my company. I'll put them in one bucket, even though we differentiate in certain ways. I'll talk about that. And then like a Jonah and then a Viome. Yeah. But I'll then distinguish just to make this easy. So with GMAP, the key technology they're using to really explore the microbiome is a PCR. PCR is cool, it's informative, but in the context of the microbiome, it can tell you much. When you're doing a PCR, you build what we call primers to identify specific things you're looking for. You only have a pre-selected panel that you're looking for. They're looking for 12, 13 organisms, and then build a primer to look for those organisms. But the problem with that is you have hundreds and thousands of species in your gut ecosystem. You cannot actually see the other key players in your gut ecosystem that are either contributing to your health, your symptom, or the disease that you're having. So that's one. The other challenge with that is that with the PCR test, you don't have insight into relative abundance. You only have insight into absolute abundance. So relative abundance is I look at my gut, I know which organism takes up what proportion in relation to others in that space. With GMAP, it's absolute abundance. How much of this organism did I see? How many CFUs of this organism did I see per gram of stool? How much of this organism did I see per gram of the sample that I collected? Now, in the context of trying to understand the microbiome and gain insights into dysbiosis, that it makes it difficult to understand balance. If there's a synonym that comes to mind when you talk about the microbiome, it's balance. And you can't get a sense of balance when you're not looking at relative abundance. That's why a lot of the clinics we're selling to right now have been using the standard legacy GMAP test for 10 years. The other company that I will talk about before I talk about ours is Viome. What Viome is using is metatranscriptomics. This is a fascinating technology. The problem is it is not the best technology to probe the microbiome routinely. And I'll tell you why. The way I think about that technology is like you're building a spaceship to go to work when you can literally take an Uber. It's not ideal for the microbiome. With metatranscriptomics, what you're measuring is the gene expression profile, RNAs. If you eat, if you have a snack now, and then I profile your gut microbiome and I measure your RNA transcript level, it's going to be different versus later this evening. So although you're getting that gene expression profile, it's transient. It doesn't tell you whether these are the genes specifically encoded in your gut ecosystem. So that's one. It's very unstable. But the other bigger problem is that gene expression profiles usually do not correlate with the proteins that have been translated eventually. So you might see an expression profile for a certain protein, and it would be misleading for you to then think that RNA would eventually be expressed to that protein because we have seen in several studies that's not essentially the case. That's why a lot of people feel very disappointed when they do the test because unfortunately, it doesn't give you that strong insight. Lastly, about that, it does not allow you to tell the difference between what is constitutive versus what is transient, which is what the gap we cover with a metagenomic testing. With a metagenomic testing, we don't tell you, oh, this is the snapshot expression profile. We tell you what is encoded in your gut so that you know how to shift them and in what direction. I have said that I'm bucketing Vitract, Vibrant, Zuma, and Jonah. We differentiate. With Vibrant, I think Vibrant is actually a good test. The key difference is that Vitract has spent a lot of time to give you strong insight into metabolite, the function. Vibrant does a lot of work in showing you taxonomic profiling. It's like they can show you 200 species and all that. That in itself is also not very actionable for you as a person, even though it gives you that information. So I think Vibrant is doing fascinating work. The layer that Vibrant has added is to give you more insight into the potential function. Are you producing butyrate? Are you producing enough serotonin? Do you have species that produce enough GABA? Do you have enough species that are producing myrocyanins or urolithin, etc.? The other layer that Vitract has added is the recommendation section. When we started building this test, we had three things in mind. We wanted to build a comprehensive test, an affordable test, an actionable test. It has to be comprehensive, but you have to present it in such a way that it will make sense to the clinician. Two, it has to be affordable. If it's not affordable, you can hardly get longitudinal data because who's going to pay for it more than once? All right, this is one way that Vitract also separates from the Vibrant. We are more affordable. But three, it has to be actionable. I personally think that Vitract actually trumps a lot of the companies that way because if you look at our report, we show you things you can do. And then we show you the literature that actually back them. One thing we have opened ourselves up to is feedback. I'm pretty sure the first time we had a conversation, we also mentioned that feedback, we're always happy with feedback because that gives an opportunity for real scientific engagement. Jonah is a new test. The only thing Jonah shows you is all the organisms in your gut. There is no context to functional profiling. There is no context to how you can shift your gut. There is no context to diversity. I genuinely think it's overpriced. I don't see a reason why any microbiome test should cost that much money. There are cons to these things. I want to talk about the cons too, so it doesn't just look like we're talking about the pros. Now, the con to a metagenomic test, for example, is one of the things that we do when we are processing your sample, especially through our computational pipeline, we have to put a threshold to what can define noise, which could be a false positive, from what actually is in the gut. In situations like that, if you don't set your threshold well enough, you're likely to fall into the trap of setting the threshold too high such that very low abundance organisms will not show up. Some people might do a test and maybe they had 0.00001% Akkermansia and it will say it's zero. That's just because according to that denoising threshold, it's actually taking that out. The second challenge with metagenomic sequencing, which thankfully we've been able to take care of, is when you give us your sample, in your stool, we also have human cells there. You have to ensure that you're preparing the sample properly, like homogenizing properly, your DNA preps are well done, so that the only thing you're reporting are just the microbial population in there. I think that's one of the things that we ensured with our lab partners to do something really rigorous before we actually started using the test. So this is how those tests differentiate.
Got it. Thank you. Kasim has asked some specific questions. They're all very good. Maybe if you can just go through these quickly. I'll just read them off quickly, and then if you can give me your quick take on each of them. Serum versus stool zonulin. Let's start with that one. Serum versus stool zonulin.
Stools on the lean can be very misleading because there are other families of proteins, like complement proteins, that when elevated in you, it can actually show up as though you have high zonulin, but you don't. Secondly, there are studies that have actually shown that people that even have high zonulin levels don't necessarily have increased intestinal permeability. What I would encourage in that context is you can be looking for other biomarkers like LPS producers, like the proteobacteria, because they are known to actually increase intestinal permeability. And when you have a leaky gut and the LPS actually escapes into circulation, we have seen in studies also how serum LPS actually correlates with fecal LPS. So that can actually be a very good indicator for circulatory inflammation emanating from the gut.
Awesome. His second question is about assessing the metabolic profile of the microbes is more important than measuring the functional markers themselves. So basically, if you measure butyrate, for example, versus measuring the metabolic profile.
The example is measuring butyrate might be more important than measuring the organisms that produce it.
Kasim?
Can you hear me?
Yeah, we can. Is that accurate?
Yeah, no, what I was asking was because like did some training of microbe and they argue that checking the, for example, butyrate producing species or microbes is more important because it's used. You don't necessarily have a good indication. But if you're just looking at the microbes that produce butyrate, then you don't know if they're producing butyrate. So I feel like they're both imperfect in their own way, but I was wondering if there's anything else that you would add there.
It's a fascinating question. That's why I said the sweet spot is metagenomics and metabolomics. Because metagenomics tells you about the functional potential to produce butyrate, that is the species. Metabolomics actually measures the butyrate for you. Think about this in the context as a clinician who's actually trying to make changes. If you do a test for someone and they have low levels of butyrate by, let's say you did a GCMS, which is a metabolomic test, so you actually measure the butyrate. The levels of butyrate is low. What would you do? You probably supplement with a sodium butyrate, hydroxybutyrate, but that's a band-aid. Rather, if you have data, you can actually support them more continuously by trying to increase the species that produce the butyrate. So that when you throw in dietary fiber, you're certain that you have enough species that can actually ferment them to produce butyrate. So that's how I think about it. So it depends on the question that you're asking. If it's in terms of moving things, then metagenomics.
Is there any role for sodium butyrate supplementation? Because like it is something that like some doctors that are out there, some experts, so to speak, advocate for. But in speaking with you, I'm not sure. Well, what are your thoughts on that?
Yeah, I think there's definitely a role. It would give you most likely temporary relief. It's not good. The way to go is increase the species that will produce that butyrate. And the good thing with butyrate is that your colonocytes pick them up and they actually build up to 1 to 5 millimolar, actually, in your cells. So they have the tendency, because of that buildup, to even do things like epigenetic changes like HDAC inhibition. So sodium butyrate, to your question, yes, but it will give you temporary relief. The more permanent relief is increased butyrate producing species. And there are key ways to do that. We know how to do that. First, we need to repair the gut lining, because when you repair the gut lining, you ensure that you're keeping the luminal content hypoxic. Those organisms can only grow in the absence of oxygen. They are obligate anaerobes. So that's the first thing to take care of. That's why sometimes we encourage L-glutamine or zinc to help repair that gut lining. But then the second thing is we just throw in foods that specifically enrich them. An example, Faecalibacterium, we know the things like kiwi fruit and Forcameza inulin, and things like intermittent fasting increases acamenzia more, things like exercise or things like wheat barley. These are all organisms we know have been recapitulated by more than 5 studies to increase specific species. So that's one of the ways to do that.
Awesome. And then I guess one of the last questions here, Uma asks about Tiny Health. Any thoughts on Tiny Health as a
Yeah,
microbiome test for kids?
it's a good company. I think when I think about what we're doing, I think about the closest to what we're doing to be Tiny Health. They're using metagenomic sequencing. The only difference is what Tiny Health is doing is metagenomic sequencing, but it's shallow shotgun. It's 3 to 5 million reads. We are doing 10 million reads. The meaning of this is that when you have super low abundance of organisms, we are able to pull them up. That's because we're just doing a deeper run versus Tiny Health. But it doesn't take away from how incredible what they're doing is. So I love the company. I have done tests before, so it's a good company. Secondly, we provide way more insight into what to do, like the foods, the prebiotics, and that's also because I think that's one of the main theses of us starting the company in the first place. That's the superpower we have, but I do consider them to be a good one.
Awesome. I know we've gone over the 30 minutes. I think we should probably make these an hour going forward. And I think you've answered all the questions here. If anyone else has another question, put it in the next 20 seconds. Otherwise, I'll just say thank you so much, Chidozie. I met Chidozie a few months ago, maybe earlier this year, and we spoke a little bit about ViTrack. But beyond that, I was just so impressed with his approach to the microbiome. And I think that what we are about here is education and giving people that foundational perspective to help them navigate these complex and evolving fields. I really appreciated the perspective you shared with me back then, and I think that really came through today. The name of your test is ViTract. What's the best way for people to engage if they're interested in trying it out? Do you have a suggested way to do that?
Yeah, they can just go on vitrack.com. If you're a clinician, create a practitioner account so that you can get like an incredibly much lower discount. For one of the tests, we just sell directly to practitioners. For the first test, 16S, we sell both direct to consumer and to practitioners. So just create an account on vitrack.com.
Awesome. Thank you so much. Is ViTract available globally? Where is it available?
As of today, US and Canada.
Okay. Are you working on global yet, or?
Yeah, we are getting a lot of demand from Europe, so
Yeah.
we're trying to work out distribution there.
Okay, awesome. Thank you so much, Chidozie. This was wonderful. And thank you all for those of you who logged in. I will make the recording available. You do have to be part of the membership to see the recording, but I will make the recording available to people who are part of the group and create a text summary of what we talked about. Thanks so much for joining, and I hope you all have a great weekend.
Thanks for having me, Anand.